My enzyme activation Play-Doh modeling. So first you start with the enzyme. It's got an active site right here and here, and it's combined with the substrate right here, and that's going to form an enzyme substrate complex, which looks like that. And this is a lock and key model, but if we're going to do the induced fit model, the substrate or the enzyme is actually going to mold to the substrate. That's going to look something like that. It's just going to kind of like fold around it. And then what the enzyme does is it weakens the bonds in the substrate and breaks it into two pieces. An example of this is with the enzyme amylase. It breaks starch down into sugars inside of your cells, which then you can use as energy. This is my model for competitive inhibition. So we've got our enzyme, we've got our substrate, but now we have a competitive inhibitor up here. And what that does is it bonds to the active site of the enzyme so that the substrate can't get in there. And now what this does is it keeps you from having too much of your substrate getting catalyzed by the enzyme. And if you increase your concentration of your competitive inhibitor, then you're going to regulate how much of this you have being catalyzed. And if you decrease the amount, then this will increase. If you increase the amount, then the amount of this stuff catalyzed will decrease. An example of this is sulfanilamide. Acts as a competitive inhibitor for para-aminobenzoic acid, which and it does this by binding to the active site of the enzyme that it normally binds to. Another type of inhibition is non-competitive inhibition. And this occurs when the inhibitor is not the same shape as the substrate and is not similar in form, but there's another active site on the enzyme that it can bond to down here. And by doing so, it blocks the active site for the enzyme. See, now it can't fit. An example of this is in protein synthesis. So the substrate is RNA polymerase, and if the repressor gene is there, then you cannot have any RNA polymerase. Our protein synthesis doesn't start because there is no RNA polymerase being bonded to the enzyme. Another example of enzyme regulation is allosteric inhibition. And what happens in this is there is two active sites on the enzyme, and we've got our enzyme, our substrate, and our allosteric inhibitor. And the enzyme, or the allosteric inhibitor, bonds to the active site of the enzyme. And what this does actually is it changes the enzyme's shape so that it is no longer usable by the substrate. Now, an example of this is in pyrimidine, the nucleic acid pyrimidine, and the synthesis of that. ATCase binds a CTP to create that, but if you have enough ATP, it stops that process from happening. A subset of allosteric inhibition is called cooperative inhibition. And this is really interesting because the inhibitor actually opens up more active sites on the enzyme that then can be bonded to by the substrate. And in this example, we're using hemoglobin and oxygen. And when one oxygen bonds to the active site of hemoglobin, all these other active sites, they open up. And now we have 3 more active sites that we can all bond oxygen to. And that is about cooperative inhibition.